In the previous episode, we explored mitochondrial DNA, the DNA passed from mothers to their children. Every one of us carries a small piece of our maternal history inside our cells. Today, we're exploring the opposite side of the family. Instead of following mothers, we're going to follow fathers. The Y chromosome has become one of the most powerful tools ever developed for genealogy, archaeology, anthropology, and human history. Unlike the other chromosomes in our genome, the Y chromosome changes very little from generation to generation. Because of this stability, it serves almost like a historical document that has been copied continuously for tens of thousands of years. Every son inherits nearly the exact same Y chromosome as his father. His father inherited it from his father, and so on, generation after generation. Eventually, every surviving paternal line converges upon one individual. Scientists call him Y-Chromosomal Adam. But, as we'll discover today, he wasn't the first man. He wasn't the only man, and he wasn't even alive at the same time as mitochondrial Eve. Humans normally possess 23 pairs of chromosomes. 22 pairs are called autosomes. The remaining pair determines biological sex. Females generally inherit 2 X chromosomes. Males inherit 1 X chromosome from their mother and 1 Y chromosome from their father. That simple fact makes the Y chromosome incredibly useful. Because only fathers pass it to sons, the Y chromosome usually follows the same path as surnames in many cultures. If we trace the paternal line backward, from father to grandfather to great-grandfather, and so on, eventually we're looking at the exact same chromosome carried centuries ago. Although mutations slowly accumulate, most of the chromosome remains unchanged. This makes it possible to reconstruct family relationships extending thousands, and sometimes tens of thousands of years into the past. Most chromosomes undergo a process called recombination. During reproduction, your mother and father's chromosomes exchange pieces with one another. Think of it like shuffling 2 decks of cards. Every generation creates a new mixture. The Y chromosome is different. Most of it does not recombine with the X chromosome. Instead, it passes almost intact from father to son. This means mutations accumulate one at a time. Those mutations become permanent historical markers. Scientists can identify exactly where those markers occurred and determine which men inherited them. Every mutation creates another branch on humanity's paternal family tree. The most important mutations for deep ancestry are called SNPs. SNP stands for single nucleotide polymorphism. This simply means one DNA letter changes. Perhaps a G becomes an A, or a T becomes a C. Most SNP mutations that define Y DNA haplogroups are believed to have arisen only once, and were then inherited by all descendants of the man in whom they first occurred. When that mutation is passed to all future descendants, it becomes a permanent signature of that ancestral line. Imagine a man living 30,000 years ago experiences a new SNP mutation. Every son who descends from him carries that mutation. Every grandson carries it. Every descendant today still carries it. That single mutation identifies an entire branch of humanity. Modern Big Y testing identifies tens of thousands of these markers. The more SNPs2 men share, the more recently they share a common paternal ancestor. Another type of mutation used in genealogy is called an STR. STR stands for short tandem repeat. Instead of changing one DNA letter, a short sequence repeats multiple times. These repeat numbers slowly increase or decrease over generations. Unlike SNPs, STRs mutate relatively often. That makes them useful for recent genealogy. If 2 men differ by only 1 or 2 STR markers, they may share a common ancestor only a few hundred years ago. Before Big Y sequencing became widely available, STR testing was the primary tool for surname projects. Today, STRs remain useful, but SNP testing provides far greater precision for deep paternal history. As SNP mutations accumulated through time, humanity's paternal tree continued branching. Scientists organize these branches into what are called haplogroups. A haplogroup is simply a family of related paternal lines. The oldest branches formed in Africa. As humans migrated around the globe, new mutations appeared. Each migration added another branch. Eventually, today's enormous paternal tree emerged. Every modern man belongs to one specific terminal branch. Some branches include millions of living descendants. Others contain only a handful. With modern sequencing technology, genealogists can often identify branches that formed only a few hundred years ago. One of the biggest misconceptions is that Y-Chromosomal Adam was the first human male. He was not. Thousands, perhaps hundreds of thousands of men lived alongside him. Most had children. Most left descendants for many generations. But eventually, every one of their purely paternal lines ended. Some families had only daughters. Some lines disappeared through disease, others through migration, famine, or simple chance. Only one uninterrupted father-to-son line survived continuously into the present. That surviving line belongs to the man we call Y-Chromosomal Adam. He's simply the most recent man from whom every living male inherited his Y chromosome. Perhaps the greatest surprise in human genetics is that Y-Chromosomal Adam and mitochondrial Eve almost certainly never met. These titles sound like they describe a couple. They do not. They represent two entirely different genetic lineages. One follows mothers, the other follows fathers. Each lineage experiences extinctions independently. As a result, the most recent common maternal ancestor lived during one period. The most recent common paternal ancestor lived during another. Current evidence generally places mitochondrial Eve roughly 150,000 to 200,000 years ago. Y-chromosomal Adam appears to have lived roughly 200,000 to 300,000 years ago, though estimates continue to improve as more ancient genomes are sequenced. These dates overlap broadly, but they do not indicate that the two individuals knew each other, lived together, or founded humanity as a pair. To understand why only one paternal line survived, we need to understand population bottlenecks. Imagine a population of 10,000 people. A volcanic eruption, an ice age, disease, or famine reduces the population to only a few hundred survivors. Most family lines disappear forever. The survivors rebuild the population. Although millions of people may exist thousands of years later, their ancestry traces back through only those few survivors. Repeated bottlenecks dramatically reduce genetic diversity. The Y chromosome is especially vulnerable because only fathers pass it to sons. If a family has daughters but no sons, that paternal line ends permanently. Human history contains countless examples of paternal lines disappearing. Wars disproportionately kill men. Entire tribes vanish. Dynasties end without male heirs. Migration isolates populations. Disease removes whole villages. Each event prunes branches from humanity's paternal tree. Over thousands of generations, surviving branches become fewer and fewer. Eventually, every surviving paternal line converges upon a single common ancestor. Today, Y-DNA testing has transformed genealogy. Researchers use it to identify unknown fathers, confirm surname studies, reconstruct medieval family trees, track Viking migrations, study Native American paternal lineages, investigate royal dynasties, and even compare living men with ancient skeletal remains. Every new Big Y test discovers additional SNPs. Every discovery adds another branch to humanity's family tree. The tree is literally growing every day. Every living man carries an extraordinary inheritance, a chromosome passed from father to son across thousands of generations. Its tiny mutations have become milestones marking humanity's journey across continents and through time. Although most paternal lines eventually disappeared, one survived unbroken from an ancient African man whom we call Y-Chromosomal Adam. His story isn't the story of one man alone. It's the story of every father who came after him. And thanks to modern genetic genealogy, we're reading that story with greater clarity than ever before.